let us know something more about gastrulation in mammals. That is, we would talk about the formation of gastrula in a mammal. The first thing that we must keep in mind, blastula. Here is a layer of cells in the periphery which we have called as a blastocyst, and right in the center there's a mass of cells. We have called this as inner cell mass or embryonic cells it is, and this is the trophoblast. Trophoblast is the outermost covering, and the cavity is a blastocyst cavity. Let's name the parts. This is blastocyst cavity. As we saw, it's filled in with blastocyst jelly, and this is inner cell mass. That is, these are the embryonic cells. Now, this is what is the blastula, or it is better named as blastocyst, which has to get transformed into the 3 germ layered state, that is gastrula. Now, how does that happen? First thing that happens is the embryonic cells, the inner cell mass, it reorganizes itself in such a way that this is the outer layer of cells. This is the inner cell mass. Now, as I said, these cells are reorganizing themselves. Simultaneously, the trophoblast layer, it also rearranges in such a way that all the cells get slightly elongated. What I mean, each of these cells, this is what it becomes like. Same number of cells, but now they will be covering a larger area. That means the sphere would be slightly a larger sphere. And this is where the inner cell mass would stay. The point at which the inner cell mass is attached to the peripheral layer, it is the amniotic point. Now, it can be clearly seen in the sphere that one half of the sphere contains all the embryonic cells and other half doesn't contain any of them. That would mean one half we can distinguish as something that we earlier used to call as animal pole and vegetal pole. Now we will name it as embryonic pole, and this will be embryonic pole. embryonic pole and abembryonic pole, and hence this stage would be called as a polar stage, polarized sphere. Now in the next moment, these cells, the trophoblast cells, their intercellular membranes, they disappear. So what you see there is almost one single cell with all these nuclei. That would mean it is a syncytiotrophoblastic blast. That is a multinucleate trophoblast. Embryonic cells are still in the same position. In the next stage, these embryonic cells, as I said, they start rearranging themselves. This is, say, the peripheral layer. These cells they arrange themselves into two distinct layers. This upper layer, we will call it as epiblast layer. This lower layer, we would call it as hypoblast layer. Epiblast is going to be the future ectoderm. Hypoblast is going to be the future endoderm. So, suppose if we focus only on this part. This is ectodermal origin. This is going to be the endoderm later on. Now there's one particular point in this covering from where a few cells start modifying themselves. From here, a few cells get released and they give rise to one more layer in between, and some of these cells, they are released into this cavity. Now, as I said earlier, this is going to be the ectoderm. This is going to be the endoderm. And the new formed layer, as you can guess, is the mesoderm. But as I've said, some of these mesodermal cells are released here. That means in this diagram, they will be released here. They would be forming one continuous layer inside this trophoblast and one continuous layer outside this ectodermal layer. This is extra embryonic mesoderm. This is embryonic mesoderm. Now, what forms here? You can just consider. This would be a continuous layer. What forms here is a yolk sac. But then you must remember that human egg cell or mammalian egg cell for that matter is alecithal. It has almost zero amount of yolk, which would mean though there is such a large prominent yolk sac, there is no yolk to be contained in it. So it's a vestigial or non-functional yolk sac that is found here. Now what you are seeing here, it's presence of all three germ layers. There's ectoderm, there's mesoderm, there's endoderm. So the outer covering, if you imagine along with it, what you see here is a mammalian gastrula with all the three germ layers being present there. Each layer has a specific fate, specific organs, specific structures of the body to be derived from. Ectoderm may be the outer covering, that is epidermis, and nervous system. Mesoderm into connective tissues, endoderm into epithelium and digestive tract. That is how they will be developing into future organs. That's about gastrulation in mammals. Next, what we're going for is something more about coelom. Coelom is the true body cavity. What is true body cavity? True body cavity is the one which originates from mesoderm. So it's called as mesodermal body cavity. Now, if this mesodermal body cavity is formed by splitting of the mesoderm, that is, one layer is there, mesodermal cells are there. If it splits into two, that is, if it splits into two, and if a cavity is developed between them. Mind well, when I am talking about a coelom, a mesodermal body cavity, I am talking about that body cavity which is covered by mesoderm on all the sides. It is, that cavity is exclusively in contact with mesoderm. Only then it's to be known as a coelom or a true body cavity, true coelomic cavity. That is what is seen here. The mesoderm is split into two with the cavity developing in between the two layers. So now that's going to be a true coelomic cavity. But when this happens, mesoderm splits into two by enclosing a cavity in between, that is called as a schizocele or schizocele. Schizocelic cavity, you can say. Whereas if it happens, like we discussed in mammals, if there are two layers of mesoderm derived and if they enclose a cavity in between, there are two different mesodermal layers are derived, and if a cavity is developed between, then that is called as an enterocele. Actually, in enterocele, what happens? One of the mesoderm has originated just in the vicinity of the gut, the alimentary tract. The other one is just inside the ectoderm. Then that becomes an enterocele. So these are the two types of coelomic cavities based on the origin in the embryonic developmental stage. But when I talk about the types of coelom, see, there are animals in which there could be only two germ layers, ectoderm and endoderm. No possibility of any coelomic cavity in there. But if we talk about flatworms, platyhelminths, there are all three germ layers there, but still there is no coelomic cavity. Such animals are called as acoelomates. Triploblastic yet acoelomates. All three germ layers, ectoderm, mesoderm, endoderm, all the three germ layers are there, but mesoderm has not split into two layers to enclose a cavity in between. That entire cavity is filled in with some mesodermal tissue. Then it's called as an acoelomate. Whereas in ashkelminths, nemathelminths, roundworms, there is a cavity. Suppose if I have to represent, this is endoderm, this is ectoderm, this is mesoderm. There is a cavity here, but that cavity is not covered by mesoderm on all the sides. It is covered by mesoderm only on one side. The other side it's in contact with endoderm, and that too it is an incomplete body cavity. Then it is called as a pseudo coelom, false coelom. Whereas what we see in higher animals, right from annelida onwards, mesoderm has split into two layers, either by enterocele procedure or by schizocele procedure, but there are two distinct layers of mesoderm enclosing a cavity in between. Only then that is called as a coelom, a true coelom, a true body cavity, a true mesodermal body cavity. These are the types of animals on the basis of the type of body cavity they develop. I'm sure you must be liking the video. So click a like, subscribe to the channel, share these videos amongst your friends and family who may be interested. Don't forget to post your comments, suggestions, queries, and keep watching them. Thank you.